Okay guys, I'm going to attempt to explain oxidative phosphorylation to you today. We've been alluding to oxidative phosphorylation as we started talking about aerobic respiration. Before we get into the technical details of oxidative phosphorylation, I just want to remind you about two different principles that you might have learned about in life science a long time ago. The first one is passive transport. Passive transport, remember, is when molecules move from a region of high concentration to a region of low concentration. This is considered passive because no energy is necessary in order for that process to happen. Passive transport, you could think about it as if you opened a bottle of perfume and the particles started diffusing out from a region of high concentration to a low concentration. No energy is necessary for that to happen. It's spontaneous. Conversely, we have our friend active transport over here, which is requiring energy. That's one of the hallmarks of it, that it actually does require energy of some form. Oftentimes it's ATP, but that energy we're going to see comes in other forms. And it requires energy because we're going to be moving particles from low concentrations to high concentrations. This is not spontaneous. This is something that actually requires energy. So we're going to see both of these processes at play here in our friend the mitochondria. Now again, the mitochondria is the powerhouse of a cell. It's where the bulk of the ATP is actually going to be synthesized. Now just to remind you of some different principles, we said that the mitochondria, it's an interesting structure. It has two membranes, and you can see by having a two-membrane system, it actually sets up a really neat thing. It sets up a charge differential on two different sides of the membrane. Here in this region here, we consider this the outer matrix, and here we're going to refer to this as the inner matrix. This region in between is the inner membrane. Let me write that out for you guys. So this region right in here, this membrane right here that we're pointing to is referred to as the inner membrane. Alright, and that inner membrane sets up a charge differential. Now what we actually have happening here in the mitochondria is that this outer matrix has a very low pH, and by low pH you guys know that we mean that there's an abundance of hydrogen ions. Conversely, here in this inner matrix, we actually have a high pH, and a high pH, remember, means that there's a very low population of hydrogen ions. Okay, and so what we've established here is kind of like analogous to your car battery. We have a charge differential on these two different sides of the membrane, and this charge differential is going to drive the production of ATP in the mitochondria. All right. Now, in order for that ATP to be synthesized, there are a series of proteins. And the proteins that we're going to be spending some time talking about are right in this region right here, in that inner membrane. Okay, and so I'm going to take a second and I'm going to magnify what those proteins would look like. Okay, so I've attempted to draw the proteins that are embedded in the inner membrane. There's a series of these proteins in the inner membrane that separate the outer matrix from the inner matrix, and these proteins are all going to work together to form ATP. Now the first set of proteins we're going to be talking about are these here that you see. All right, and this one as well, and that one. Okay, so all these ones that we're seeing here in blue are actually making up the proteins that are part of the electron transport chain. All right, that's the first step in this equation of oxidative phosphorylation. The second step that we're going to see here is going to be in this protein down here, protein 5, and that's going to be part of this process that we're going to talk about called chemiosmosis. All right. So these two processes are going to happen. The first one is the electron transport chain, and the second one is chemiosmosis. Now, they're all going to work together to take these NADH molecules and FADH2 molecules that were produced during glycolysis, the preparatory phase, and the citric acid cycle. All of these NADH and FADH2 molecules have made their way into the inner matrix. All right, and now the whole point of those molecules ultimately was for the production of ATP. Now I mentioned, I think in some of the classes that we had last week, that every NADH molecule, I said, is ultimately going to form 3 ATP. And I don't know if you remember, I said that every FADH2 molecule is ultimately going to lead to 2 ATP. Okay, so that's kind of like a cliff note version of what's going to be happening. Now we're actually going to explain the mechanism by which those ATP are finally made. All right, so let's start off first with NADH. Oopsie, let's get it there. Okay, so we said NADH is an electron carrier. We had 2 formed during glycolysis in the cytoplasm, and then we had per glucose molecule, and then we had 2 made during the preparatory phase in the mitochondria, and then we had 6 formed during 2 different turns of the citric acid cycle in the mitochondria. Okay, now these NADH molecules need to form ATP. Now what actually, we said it's an electron carrier. It's been storing these electrons, and now it's going to be passing these electrons on into the electron transport chain. Now what happens here is that NADH specifically matches and binds with this protein first, the first protein in our complex that we're going to be talking about in the electron transport chain. Protein 1, sometimes people just refer to it with a Roman numeral 1, is actually an enzyme that's called NADH dehydrogenase. And we know that the role of a dehydrogenase, of course, is to remove a hydrogen ion from a molecule. And it's going to be doing that to our friend NADH. So what happens specifically is that NADH is going to, actually let's draw it up here, NADH is going to find and recognize this enzyme here, NADH dehydrogenase, and as it finds and binds that protein in the electron transport chain, it actually is going to become oxidized. And as NADH becomes oxidized, it forms, remember, NAD+. All right, now NAD+, it's done. The electron that's been passed on into this protein, protein 1, NADH dehydrogenase, is now going to pass that energy on. Now as it fires, what I think of is protein 1 here is a proton pump. Now let's think about what this means, is that we said that in this outer matrix space, we have a lot of hydrogen ions. Remember we said it has a low pH, whereas in the inner matrix, we said that it has a very low concentration of hydrogen ions. Now what we're going to see happening here in this protein 1 is that we're actually going to see active transport. Now active transport, we said, is where we're going to pump a particle, a molecule, from a region of low concentration to a region of high concentration. And in order to do that, we needed energy. The energy that's going to be necessary to take this hydrogen ion and pump it against a concentration gradient up into the outer matrix is coming from the oxidation of NADH. So that electron that was passed on is now going to be coupled to the movement of this proton, this hydrogen ion. So as the electron is transporting energy, it's going to grab hold of this one hydrogen ion, NADH dehydrogenase, and now it's going to pump it against the concentration gradient. So in doing so, we get an extra hydrogen pumped against the concentration gradient. Now that's just the first part of the equation. Now that electron that was passed on to NADH dehydrogenase has more energy still inside of it, or to pass on. It's going to pass that energy on then to this protein, which is then going to carry that energy now to this protein, protein 3. Protein 3 is also going to be a hydrogen ion pump. Now again, we said that there's very few hydrogen ions in this inner matrix space, but there are a handful of them. It's one of those hydrogen ions again is going to be grabbed in this inner membrane, in this inner matrix, and when this protein pump 3 is activated from the flow of electrons from NADH, it's going to grab hold of this hydrogen ion again and pump it into the outer matrix space. So now we've gotten 2 hydrogens pumped against a concentration gradient using active transport. But we have more in the story. The electron still has more work to do. The electron is going to be passed on to yet another protein in the complex, which passes it on in turn to another protein complex. Now we're at protein 4. Protein 4 is also a hydrogen ion pump. Its job when it's activated from the flow of electrons is to grab hold of yet another one of these few hydrogen ions that's in this region here and take it and pull it up into the outer matrix. All right, so let's look at what we've done here. The firing of this, the oxidation of this one NADH molecule to NAD plus has now fired or allowed energy to pass from this protein pump to this protein pump to this protein pump. There was a flow here. You can see here how the flow goes, hopefully. It in turn then causes one hydrogen to be moved up into this outer matrix, causes yet another one to be moved up, and then finally a third one to be moved up. So what we've done here is we've pumped 1, 2, 3 hydrogens from a region of low concentration to a region of high concentration from the oxidation of that one NADH molecule. Now that's great. So now we've done all this active transport, and you know that active transport often goes together with passive transport. So now we're going to get to the next part of the equation, which is chemiosmosis. So now we've done active transport. We've helped to exacerbate this differential between the outer matrix and the inner matrix. And so now what we want to happen is passive transport. We don't need any energy to be utilized anymore. We now want to make energy from this. In order to make energy, these hydrogen ions that have been pumped up into this inner membrane space, this outer matrix, is now going to fall passively back through a very specialized protein here. This protein here is referred to, this one in green, protein 5 sometimes it's referred to. Its name is actually ATP synthase. So the job of this protein here is literally to synthesize ATP. Now we know we've talked about ATP in the past. We said that ATP, adenosine triphosphate, is the charged form of the chemical energy form of cells. It needs 3 phosphates in order for that molecule to be charged and able to do work. So now we're going to get that energy finally. Now you know that ATP goes together with when it loses its phosphate, it becomes ADP, adenosine diphosphate. So what's going to happen here is that we have all these adenosine diphosphates, which are like uncharged molecules that are sitting around. Those ADP molecules now are going to be recharged. They're going to be recharged, and they're going to then be, of course, converted into ATP. A phosphate is going to be added back onto those molecules in order to become ATP. Now, where does that energy ultimately comes from? It comes from the passive transport of these hydrogen ions falling back down through ATP synthase. I sometimes think of a water wheel. You guys know, if you've ever been to a old-timey place that has like a water wheel going, that the water is going to fall through, and as it's falling through, it's actually turning a mill crank, right? And as it's turning that mill crank, it's doing work. And literally the enzyme here, ATP synthase, is very reminiscent actually of a water wheel. The structure of it and everything that's associated with it, it really does, it looks like a water wheel. So a hydrogen ion is going to fall through, and as it falls through, it's going to turn this crank, and as it's turning that crank, it's doing work. The work then is coupled and takes an ADP molecule and forms ATP. So what we see every single time that a hydrogen ion falls passively through protein 5 ATP synthase, we are going to synthesize 1 ATP. And remember what we said before is that every NADH is ultimately, we said, going to form 3 ATP. And hopefully you can start to see why it forms 3 ATP. We said that every NADH pumps 1, 2, 3 hydrogens against a concentration gradient. Now these hydrogen ions that have been pumped against the concentration